Okay, let me tell you a little about this project I headed up this summer called Digital Bacillaria. Here's the GitHub repository for the project. The project is meant to be open, so that means that we have a lot of our analysis and our code and tutorials and all the write-ups and everything there on the site. Anyone's welcome to contribute in any way they want. 6 people are involved in this project so far. Myself, Dr. Richard Gordon, Ujjwal Singh, Asmit Singh, and Paras Mahan. Those 3 gentlemen are from India. They're from IIT Delhi, and they did a lot of the heavy lifting in terms of the machine learning. We also have Thomas Harbeck, who's a retired professor from Germany. He's interested in microscopy, and he's even trying to cultivate Bacillaria as we speak. So this is the genus Bacillaria. Depending on what taxonomist you talk to, there are either 1 or 2 species of these algae. It's a type of diatom, so they have very interesting properties biologically. But you'll see a lot of times Bacillaria spa, so that's the same thing. It's just a general, there's no real difference between the species in terms of the general morphology. So let's review that morphology. In 1 and 2, you can see this long rod, and you can see that this rod is kind of crooked along its surface, and that's because it consists of a number of filaments, and these filaments are cells, and they articulate against one another. So nearest neighbors to one another, so these 2 and these 2 and these 2 and so forth, can articulate against one another and generate movements through the water column. And so you can see when it's folded up, it looks like number 3, so it's a parallel array of these cells. In 4, you have some displacement of one or a number of cells, and again, one cell moves against its neighbor to produce this movement. And you have a number of different modes of motion in 5, 6, 7, and 8. They move through the water column in various ways. People don't really understand why they move, other than your basic issues of motility, as they're found in freshwater and saltwater regions. And people have some idea, there's some hypotheses as to how they generate the movement, but it's still unclear in some ways. So that's one of the things we're trying to get out of this project. So again, you can see a close-up of the Bacillaria colonies. In 27, you have movement in one part of the Bacillaria colony. These 2 cells are moving against one another, and they're taking its neighbors with them. 28, the array is spread out a little bit, so the cells have moved against one another and kind of spread out. But as you can see, they're independent filaments. And 29 and 30, these 2 panels are a close-up of these filaments, and you can see they're very complex on their surface. And so here's the space in which these 2 filaments will move laterally. And again, we have some idea of why they move and how they move, but again, we still, there is a gap in the literature there, and our project will hopefully inform that. So the goal of this talk, and the goal of our project so far, has been to segment images of the Diatom Bacillaria. And so you can see from this image, we've done this through creating these purple rectangles, and they are basically outlines of each of these cells. You can see that these cells back here are pretty blurry, but these are not. These are less blurry, and we were able to classify these, or at least identify these segments. And so these images are not the greatest images in the world. They're from YouTube videos and other materials from ranging from amateur movies to a little bit more professional movies, but using light microscopy mainly. And so our data is, we need to have a robust method. So we're going to try various approaches and evaluate the results of different methods. So back to that in a minute. But first, what are the fundamental features of a Bacillaria colony? And I say that not in terms of a biological question, but more of a machine learning question. And so in machine learning, features refer to things that you can put numbers on, variables, things that you can measure. So in an image, you're going to find features, you're going to define them, and then find them, and then that's going to be your quantitative data. So where our unit of analysis here is the single cell, or single filaments, as we showed back in the picture where we had the purple rectangles. So we know a couple things about this unit of measurement. First of all, single filaments articulate against its neighbor in the colony, and we've shown that in the images. All cells coordinate their movements to produce collective movement, so they coordinate. They're not like bird flocks or fish schools, but they do coordinate in some way to produce collective movement. They don't just move all independently. Again, we don't really have a good idea why that is, though. And then with using cells as features, we can predict the relative position within frames and movement between frames. So if you can imagine a stack of microscopy images in time, a movie of microscopy images, each frame represents a point in time. And we can take within that frame, we can take positions of different cells, landmarks on each feature, which is a cell, and then measure those where those cells are in the colony. So we can identify individual cells. We can identify position. But then if you take a number of frames and you look between the frames and you find relative position on each frame, you can predict movement. So you're taking a derivative of the position and you're able to predict movement. So we tried two approaches. And the first approach we tried was the unsupervised approach. And this is analogous with standard image processing. So people who use ImageJ are familiar with these types of algorithms. And so the algorithm, the basic process here is you take an algorithm, you apply it to an image. You can apply it to a single image, you can apply it to multiple images. But in any case, you use heuristics or simple rules to find the boundaries between regions of an image. So there are changes in their fluctuations in contrast or fluctuations in color, and those are the things that maybe are defined boundaries in the image. And so you can use heuristics instead of just kind of pinpointing them by hand to do this efficiently and do this over and over again in a replicable manner. That's basically the idea. So imagine that you were sitting in a chair behind, and you had a neutral background behind you and you had a picture of that. How would you define the edge between yourself and the background? And so you could use a number of rules to say, well, you could use the background color and just say everything with that color is the background and everything that isn't is the person, except that that color might be replicated in the person somewhere, and the background may be very heterogeneous. And so you need to have, you can use a heuristic to find the boundaries, and sometimes it's very easy, but sometimes it's not. And then also in this type of approach, we want to segment images into regions by finding the most likely edges, but not necessarily the ones that define the feature of interest. And I think you'll see in some of the pictures coming up that sometimes there's noise or there are things that confound the image that that the algorithm locks onto and defines it as an edge, whereas it's not actually an edge. So those types of false positives are a problem. And then one advantage to the unsupervised approach is that it can be done for single images. It's distribution free and doesn't require any a priori information. But again, that may be a problem because you may need information about the image to process it. So we chose two unsupervised algorithms, the first being Canny Edge, and the second being the watershed algorithm. And so, again, we didn't use a training set or labels for this. This was just the algorithm being applied to the image. And so in the left, in the Canny Edge example, you see that it wasn't very good at detecting either the colony boundary nor individual cell boundaries, although it was somewhat successful at detecting the colony boundary where it was clear, but in some cases it was not clear. And so this is a problem, and it's not easily solvable by just cleaning up the image. And then on the right you see the watershed algorithm, which is where you see a little bit better resolution in terms of some of the cell boundaries and then the edge of the colony. But again, there are a lot of gaps, and it's not really useful. So, like the canny edge detection uses a number of tricks to find an edge, and the watershed algorithm is actually based on something out of geology, where they try to find watersheds in a landscape and where those watershed limits are. And so they use those type of rules and apply it to image processing in general. So they're actually pretty good algorithms sometimes, but in this case it did not work well. So the unsupervised approach, we can basically say, is not very accurate for our purposes. The target features, the cells, cell segmentation by landmark especially, which is to say that the edges of the filaments or the cells, we wanted to get those edges and have the segments basically be just each filament and nothing else, was not attainable. So this was not attainable using this method. The watershed approach, in particular, does detect boundaries between the background and the edge of a multi-cell colony, but that really wasn't the feature of interest. And in any case, it doesn't do it perfectly, so there isn't really a trade-off there. Also, we notice in the unsupervised approach, it's really dominated by noise from the background, in particular algal masking. So there are other forms of algae that grow on top of the, or grow with the Bacillaria, sometimes in your sample because it comes from a water source. There are all sorts of things that are moving around in there, or stationary objects that get in the way. Plus, you have water, so there is some problem with getting a clear signal. And so now we move to the supervised approach. So now we're kind of getting into a little bit more complicated technique, or set of techniques. And so now we're going to do feature selection using a training set. And so in this case, we actually used a series of landmarks that define the edges of of each filament. And so here's some of the images that we used as an example, just to show what we're trying to segment. So these images were pre-processed for this purpose, and then this set of rules were applied, but they're very specific rules that define the feature. It's not done by hand, but it's also not just done blindly. And so our supervised approach, we use deep learning. So we kind of went right for the jugular and used deep learning, which of course is the reason I say jugular is because it's a very advanced technique, and it's also something that maybe sometimes is too powerful for some problems. But we use TensorFlow, which is a machine learning library developed by Google. And specifically, is the images I'll show you are being analyzed using DeepLab version 3, which is a form of semantic image segmentation, which is basically image segmentation using labels that have some meaning to them. We're also working on a tutorial for MobileNet, which is another TensorFlow library which specifically maximize memory usage. So if you're interested in this, we'll have 2 ways to do this in TensorFlow. So let me talk a little bit about deep learning for those of you who are uninitiated. So there's a difference between a simple neural network and a deep learning network. And so the simple neural network, as I'm sure a lot of you are familiar with, has an input layer where you input labels and data, numerical data. And then there's a hidden layer where all these things are processed and sorted in a combinatoric fashion. And then the output layer puts it together and puts every piece of data into a class, or classifies things. So the idea of a neural network is you put your data in and you get an output. You update the weights between the input layer and the hidden layer, and the hidden layer and the output layer, and you end up with your data fitting into one of 4 classes here. So that can get you, so far. But deep learning, on the other hand, actually is a bit more complicated. In deep learning, you have multiple hidden layers, hence the word deep. And these layers actually contribute to the ability of the algorithm to search for a solution, a more complicated solution. There are a lot of, there's some problems with error, and there are some problems with underfitting and overfitting, but those are, problem specific. But in general, deep learning does solve really hard problems better than a neural network. And it's interesting because the neural networks were first proposed in the 40s and 50s. And then, it took a while for people to come up with a backpropagating neural network. That was in the late 80s. And then it took all the way to 2012 in order for people to figure out how to do deep learning. And this person here, Jeff Hinton, he's one of the pioneers of deep learning, and he was the one who really kind of pushed it to its breakthrough phase. And so it's become a very hot area. And again, people apply it to many, many types of problems, and maybe often problems that don't really deserve the deep learning approach. And then the difference between normal machine learning and deep learning, as a point of contrast, is that you'll take an input and you need to do feature extraction and classification. So in machine learning, feature extraction is somewhat manual. So you have to kind of define the features and guide it along. You might have to provide a mask that you've created by hand, and then use a classifier and then get an output. With deep learning, you can take an input, do feature extraction using some set of rules, as we'll show, and then you continue on to classification. So those things are included in a single model, and then you get an output. And so there's a blog post here that is the data scientist, what deep learning is and isn't, where I took some of these images, and you'll probably want to read about it if you're interested in this topic. There are other resources as well, and they should be on the DevoWormML resources page on GitHub. So deep learning is, our strategy for deep learning is where we define the features of, landmarks for these features. So, it might be like where the extreme edges are of the filament, the sides, how wide the filament should be, and then a centroid of some type. So if we can define those things and say to have the system find these things in some of the candidate features, then we'll be in a better position to actually define features more precisely. And so we've defined the landmarks on features. We define the training set, which is a set of images that we want to train the system on. So we have a set of images that we pre-processed and cleaned up and give the system to draw these purple lines on. And then the results should be some sort of bounding box that defines a cell. So we're identifying the feature with this bounding box. And so here's some preliminary data analysis. So in this case, this image on the left is represented by these dots. So this image is processed, and then an XML file is produced, and then we can take data from the XML file and plot it on a bivariate graph. And in this case, we have a number of different components of each filament. So we have the corners of the bounding box, so the upper right and left-hand corner, the lower right and left-hand corner. And then, so the X and Y components of the bounding box, and then a centroid for each bounding box. So the centroid is the midpoint between the ends and halfway between each side. And so this graph is a little messy, but it's all of those points sort of amalgamated into a cloud. And you can see that cloud, if you turn the image in the right orientation, matches up. So you can see the colony here in this image. You can see the point cloud in the image. And this scale is not really regular. It's just something that's produced by the algorithm. It just takes the image at a certain point and counts how many pixels over it is. So this scale doesn't mean very much. And we can do it with another colony. And this colony is a little bit different shaped, but basically they're the same types of points are plotted on this graph. And again, the scale is not important so much, but you see, and it doesn't look very good at first, but then you see that you're dealing with the corners of the bounding box and a centroid, that you can kind of see the shape. Now we don't, we're not just interested in like dividing things out of the shape of a point cloud. That's not really useful. We do want to get a sense of if it fits the data, though, and that's why I did this exercise. What we really want to know, though, first of all, are these points consistent with the images? And they seem to be. There are other tests we can do, but also if we take a stack of images in time and figure out the series and figure out if we can get the points out and then normalize the scale, can we draw a straight line? Can we predict motion? And that's sort of the next step here. I'm not getting into that right now, but so just another example where we have points representing the centroid of each bounding box only. So this is a clearer view of how this point cloud fits the actual colony. So this model here fits the colony pretty well. There are a couple of crooks in the morphology, but that's probably due to maybe some measurement error, maybe just the shape of this thing. So, I mean, it's a pretty good, so far so good. I think we're in pretty good shape so far. So the results for the supervised approach are more accurate, but there are a few caveats. So we talked about the target features being attainable, which they are, but we need now to figure out a common coordinate system and to confirm the results. So we didn't do the testing phase yet. What you saw there was the training phase. And so we took a bunch of images, we put them into this deep learning system, we defined some parameters for the features, and it gave us the features. So it can classify things according to those features. But there wasn't a lot of variation in the images. There wasn't we can't generalize very much from this. The testing set, which is forthcoming, should allow us to do this a little bit more broadly. Now, can this method be used to detect shape and movement changes across frames? Can we do this just with the data that I showed you, or can we do this in another way? Maybe. We may need a metric for this. We may need to develop metrics above and beyond what just sticking it into a deep learning algorithm. And we notice that the noise has been minimized, but there's still some incongruities with the original ground truth image. And so some of this is rotating the image, but also some of it is just finding the proper scale. So, finding, making sure that things are equivalent to one another across images. And so that's the talk. Thank you for listening.